Greetings to all. In this video, we are going to see about principle of biotrope, hemibiotrope and necrotrophs. Colonization of fungal pathogen. Pathogenic fungi uses biotrophic, hemibiotrophic and necrotrophic infection to infect plant and reduce crop productivity. Traditional classification of fungal pathogen relied on the mode of nutrition. They are classified into three types. Classification of fungal pathogen: biotrophs, necrotrophs and hemibiotrophs. These are the example for hemibiotrope and biotrope. Biotropic pathogen. In this, they derive nutrient and energy from living cells. Like biotrophic pathogen extract nutrient from living cells. Most of the biotropic fungal pathogen form specialized feeding structure called haustoria to investigate host plasma membrane, deliver effector molecule and allow the free exchange of nutrient from the host to the pathogens. Biotrophs have been proposed to have evolved from symbiotant by adapting effector to evade recognition during the colonization process. The biotrophic effector plays a crucial role in promoting host colonization by suppressing host defense surveillance system. However, resistant plants recognize these effector or their action to elicit defense to ward off pathogens. Hemibiotrophic pathogen. Hemibiotroph initially invade live cell prior to transiting to a necrotrophic lifestyle to obtain nutrient from killing the host cell. Hemibiotrophs initially extract nutrient from living tissue before switching to a necrotrophic phase. Hemibiotrope pathogens are known to undergo lifestyle transition by initially adopting biotrophic lifestyle, which is followed by necrotrophic phase during the course of infection. The switch from biotrophic to necrotroph is anticipated to be triggered due to the increased pressure from the host defense and also enhanced need for nutrient to facilitate aggressive growth of the pathogen. It is becoming apparent that hemibiotrophs deploy different effectors to differentially adapt to biotrophic or necrotrophic mode. Necrotrophic fungi. Necrotroph derive their energy from dead or dying cells. Necrotrophic pathogen kill plant cell to access nutrient. The necrotrophic lifestyle of a fungal pathogen has been anticipated to have evolved from saprophytic mode of nutrition acquisition. Necrotrophs are known to kill host cells to colonize. However, some recent studies have suggested that they do not adapt a brief biotrophic phase to suppress host defense before entering into necrotrophic phase. Necrotroph utilizes an arsenal of toxin as well as cell wall degrading enzyme to destroy host cell and obtain nutrient. The toxin can be host selective toxin that are effective on particular host genotype, whereas non-host specific toxins are effective on a wide range of plant species. Induction of DNA damage, abnormal mitochondrial oxidation, cytotoxicity, etc., leading to host cell death is some of the common function of host selective toxin. Necrotrophic effectors promote susceptibility by inducing host cell death either through direct action or by induction of host defense response. In this slide, we are going to see the difference between biotrope, hemibiotrope and necrotrope. Features: feeding in biotrope on living host cell, in hemibiotrope initially on living and later on dying or dead host cell. Necrotrophs on dead or dying host cell. Obligative or facultative. Biotrope obligative, hemibiotrope facultative, necrotrope facultative. Feeding structure: biotrope haustoria, hemibiotrope haustorium-like structure in some cases. Necrotrophs no haustoria. Host range: In biotroph, narrow. In hemibiotroph, narrow. Necrotroph, broad. Hormones involved in defense. In biotroph, salicylic acid. In hemibiotrop, salicylic or jasmonic acid. In necrotroph, jasmonic acid. Effectors. In biotrop, avirulence effector, gene for gene interaction. In hemibiotrop, avirulence effector, gene for gene interaction. In necrotroph, host specific toxin, necrotropic effector. Resistant gene. In biotrop, qualitative. In hemibiotrop, qualitative, and in necrotroph, quantitative. These are the examples for biotroph, hemibiotrop, and necrotrophs. In this image, we can see primary biotrophic infection and secondary biotrophic infection. The mode of colonization of fungal pathogen in plants. Biotrophic fungus forms an apposorium to penetrate inside the host. It develops a specialized Eiffel structure called haustoria to deliver effectors into the host to suppress its defense as well as to obtain nutrient from the living tissue. In secondary necrotrophic infection, necrotroph fungus, the Eiffel penetration is facilitated by dissolution of the host cell wall by secreting cell wall degrading enzyme as well as toxin to kill the host tissue. The necrotropic fungus derives nutrient from the dead tissue of the host. The above content is taken from the article that are listed below in the reference. Thank you for watching my video. Don't forget to like, share, comment and subscribe my channel.